Okay, thanks so much, everybody, for joining. I really appreciate your time to spend lunch with me. My name is Jack DiGiovanna. I'm the Director of Programs at Seven Bridges. Been with the company for about 5 years now. And the thing I wanted to talk to you today about was human learning. So how could we train the next generation of data scientists and bioinformaticians on the cloud? I'm going to cover a few different things here. First, I wanted to introduce Seven Bridges for people who are unfamiliar with who we are and what we do. Next, to frame the problem statement of, what do humans need to learn? Why do we need to have, what are we trying to solve? The middle of the talk is an overview of the data analysis ecosystems that we have and how they were built and what they do, what problems they solve. And then the end of the talk is, how we've leveraged these platforms over the last few years as an online teaching foundation. So Seven Bridges was formed over 10 years ago. We had our 10-year anniversary last October. And it was the fundamental problem of how do two people collaborate to analyze 1,000 genome data, right? The data was already getting big 10 years ago. It was hard to collaborate. It was hard to find tools. It was hard to find compute. And over time, we've developed with both commercial and academic partners, government, private foundations, and we really took on the mission of connecting the world's biomedical data to accelerate research and improve health. We have an industry-leading collaborative bioinformatics platform. Also, something that differentiates Seven Bridges is we have really strong scientific and bioinformatics services in-house in terms of the number of PhDs on staff, the number of people who are dedicated just to customizing bioinformatics. Beyond the platform and product angles, we also have innovative analytic approaches. One of the best known ones is our graph-based aligners that we use, multi-genome graph-based aligners for genomics, and some other things in terms of machine learning that we're doing. And access, we can provide access through partnerships with different government agencies. We can provide access to data sets for folks who are authorized to access them. We've got a number of awards over the years, which is really nice to show on the right here for the things that we've built and the things that we've done. And here we have a big infographic, which has some cool animations. It's always exciting to show animations and little pills falling down into the product jars there, of where does Seven Bridges fit within the entire analysis journey. And one second, I'm just going to turn on my pointer here. So it starts on the left here really with making a hypothesis. So anyone is going to do this. And the platform sits in the middle as the foundation. So this person, she could be doing iterative analysis and a number of different things. She might be bringing in her own data or proprietary applications. Maybe it's public data and public applications. And the end of that story could be a clinical trial. It could be diagnostics, productization. Maybe there's some cohort stratification that's going on. And obviously there's no one super client who's doing all of these things, but we have a number of different clients that work with Seven Bridges to be able to fulfill different parts of these journeys. The whole thing is importantly guided by industry standards all throughout, so that we're using things like GA4-GH standards to make sure that it's incorporating them as much as possible to make sure that it's interoperable, it's reproducible, and that everything is properly described. And we serve actually, what this is doing, what this allows us to do for our clients is to optimize the product to market lifecycle for users of Seven Bridges platforms. We're actually divided as an organization into 3 different segments. So there's the commercial segment that focuses on pharma, biotech, diagnostics. There is also a national initiatives segment, which is working with larger nation-scale projects such as the Turkish Genome Project. And finally, my team is the program side, the programs team, which is working with the NIH agencies, NCI, NHLBI, Common Fund, the VA, as well as nonprofits such as Zero Childhood Cancer and the Pancreatic Cancer Action Network. So my team's mission statement would be more optimize the research to publication lifecycle, but it's the same idea. We're trying to help our users, get to more actionable outcomes and information at the end. This is a picture of my team. I highlighted in blue the people who are responsible for the platforms that we're going to show, or who actually led the teaching courses that we'll go through. I just want to give a shout out. We're also hiring program managers, and the link's on the bottom. So if you're interested and it's a fit, please get in touch. So this brings us to the problem at hand. What do humans need to learn? And this talk I initially gave for the CI4CC, the Cancer Informatics for Cancer Centers talk in the fall, of an opportunity of what do we try and train people? And what I've heard from people in professional colleagues, friends, both in academia and in industry, is that STEM graduates are not optimally prepared. And you could take the cranky side of this. And in my day, I walked uphill both ways in the snow to school, and I knew everything. But I think you could take it as a more glass half full approach and say that data and the different tools that are available and the problems to solve are just expanding so quickly. I mean, if you look at COVID, for example, nobody knew about this at Christmas. Like we barely heard of it. And now we're shifting so much development and effort to try to help respond to this pandemic. And I think it's great. But you could have never had someone who graduated in the fall prepared for coronavirus. So not only did I hear this from different friends and colleagues, but this was also an outcome of the NIH Strategic Plan for Data Science in 2018. So one of the pillars, I think this was from Figure 3, was how do we develop the workforce? So both enhance the existing NIH data science workforce, but also to expand the national research workforce. So train the people we have and find new humans to make it even stronger. So this was pretty interesting. And I reflected back to what did I do when I learned data science? Before coming to Seven Bridges, I was in machine learning or data science. So how did I approach that? Well, the first part is the math, right? So you make a model of the system. So here we took a very simple linear model. Choose your cost function. We'll use LMS. Take the partial derivative of the error with respect to the weights. The chain rule works its magic. And there, you have an update equation. And now you can do machine learning. You can learn this system. You can use a better equation, use an MLP, use something else. You get to a universal function approximator. Change your cost function, use reinforcement learning, use deep learning. All these different things you can do. And the math is really important. And this was, I thought, really key to learn. But reflecting back, it's only a small part of the job. And 15 years ago when I was really learning how to do this all by hand, now if you look at it, most of these things are already available as packages. You can even argue how much do you need to invest in understanding the math and the models versus the other parts of the story, which I'm going to show next, is once you have the mathematics underway in hand and you know what you want to do, the big problem comes with how do you find data? So you can do a lot of work on the iris data set or the handwritten digits data set and figure out how to train your models on that to make a better and better prediction. But what if you want to work in AML? What if you want to work in hypertension? How do you get to those data sets? Both getting to them from an approval process and getting to them from a understanding what the data is. Once you solve that, there's still the much larger problem, which is a much larger part of the job, is being the data janitor. Okay, why are these gene names saved as dates? Why is this entire column sparse in this one part of the data set? How do we bring these things together? And I think that's something, if you look at the right side here, different people give it different amounts, but you can argue that's easily more than 85% of the job is getting this data in order and getting it organized. So the question is, if we assume people understand the math or they can figure that out on pencil and paper or get that elsewhere, do we have any way to help with this part of the story? So I want to quickly give an overview of the data analysis ecosystems. Spoiler alert, they will be an important part of that story. But I wanted to first present what they are and what communities they serve so people understand when we say, OK, this was on Biodata Catalyst, what are we referring to? The first was the Cancer Genomics Cloud, funded by the National Cancer Institute. This started around 2014. It went live in the end of 2015, early 2016. And it is the idea was you would start with this genomics data, the Genomics Data Commons data, and put it in the cloud. Because before, while the TCGA was an amazing project, and generated 500,000 files, vast amounts of data, vast amounts of paper, vast amounts of publications, excuse me. Only certain universities were really able to access and leverage it. I mean, universities that had a dedicated HPC and a core lab around it that could help make that available and actionable, those researchers did really well. But the really visionary aspect the NCI wanted to push forward to was to democratize access to that data and make it available to a broad community, both different user personas, from the person who's writing bash scripts to the person who just wants to point and click, but also across different institutes and universities, not just at the top 10, not just at the Ivy League, but all across the United States and all across the world that could access this data. So it had the genomics data in the cloud, all the normal bells and whistles of the cloud, authorization and authentication to reach these diverse users, different ways to get to it. And over time, it's grown to include proteomics data, imaging data, canine data, clinical trials data. And it's really exciting to see how that has grown and changed over time. It's now live. It has thousands of users and millions of hours of compute. The next one that came along is Kvatica. This we started in 2016 and co-developed with Children's Hospital of Philadelphia. It's funded by Data-Driven Discovery and Biomedicine Center and the Common Fund in order to serve users of working with pediatric cancer. So similar to the CGC, it's working on cancer, but now it's on pediatrics. With pediatric cancer, you're fundamentally dealing with a rare disease just because you have so many fewer patients. So this was pooling together two consortiums, the Children's Brain Tumor Tissue Consortium and Pacific Pediatric Neuro-Oncology Consortium. And really one of the key features of Kvatica was being able to bridge together, bring in these things as different volumes so that individual researchers and individual clinicians could pool the data together to get that statistical significance to continue to address questions and drive forward care. In 2017, it was selected as the sort of foundational platform for the Gabriella Miller Kids First Pediatric Research Program, which means that it expanded to become also serving for birth defects as well, which is, again, another rare disease fundamentally. The final platform I want to discuss is Biodata Catalyst. This is funded by the NHLBI. It's a data science ecosystem to enable heart, lung, blood, and sleep research. And similar to the other platforms, it's enabling finding, accessing, sharing, computing, and secure workspaces. What's unique about this one is it's an ecosystem in that you don't necessarily have an end-to-end experience just in 7Bridges or just in the Broad or ISB or another component, but now you have a whole ecosystem to maximize functionality to users. And really, one of the things we're driving forward is interoperability between them so that you can pick and choose cafeteria style the best components and bring together the entire story. So these are three very different platforms that were built for different user groups, have different tools, have different data. But looking back at them and reflecting back, you find there's common elements for all of these analysis ecosystems that we have, all of these platforms. So collaboration is a key one. So starting with secure customizable workspaces, which is shown here. We call it a project. People also would call it a workspace, call it a sandbox, call it whatever you like, but there's an owner to it. They can invite collaborators with role-based permissions. Files live there. Apps, which could be tools, workflows, pipelines, scripts, also are part of the project. And then as you execute those things, it generates a task and new files live there again. So you have this nice container for organizing your work. It also helps if you have controlled data, so you can use that as a controlled project and only people with the right authorization can go in there. And also there is really nice billing around that you can say organize your spending within the project. So everything that happens in that project will get charged to one billing group. For example, my R01 from 2 years ago, and then if I get collaborative funding from Common Fund, I can use that and change the funding to that. The second element is usability. As was really brought forward in the CGC use case, it's really critical to be able to work with diverse users and also have workflows available to start with. Here we're showing sort of the spectrum of our interactive analysis on the bottom. So this is just a IPython notebook that's running right here. It could also be RStudio. It could be Julia. This is running within the project so that you can interactively analyze the data on the cloud, along with a number of nice usability features that make sure that the cost doesn't get out of control, that it's actually shareable. You can collaborate on these notebooks, that you have reproducibility. And if you have, RStudio, obviously, you can then get to Shiny applications. Again, serving the users who don't necessarily want to code, but here's one that's the PDX portal that just went live. Patient-derived xenografts, which is you're able to see here, a number of ways to link through, to explore the metadata, to form a cohort, which is using the CGC as a backend to do all the work. But this is able to make this data accessible to users who have those needs and fit that persona. In the middle here, we have our drag and drop visual programming interface. This is working with a Callisto tool, adding on a new tool to the end. It's kind of fast, I'm sorry, and the resolution's not great, but the tools are in teal that get dragged in. The little gray circles are the outputs or inputs. And here they're just wiring them together and configuring the tool and then saving a version of the application. And finally, you have, for people who want to write, in my opinion, pretty scary, command line scripts here that have so many flags I can't count them. That is also perfectly, the platform is perfectly compatible with that. And although we're showing a lot of things via GUI here, there's also compatible with doing this all via API as well. Reproducibility is in the middle here. This is really table stakes for doing science, right? If we want to prove something, we need to be able to replicate it. We need to be able to defend it. We need to reproduce it in other areas. There's two ways that we approach this at Seven Bridges across all of our platforms. The first is with portable and reproducible tools. Again, this could be a tool, a workflow, a pipeline, depending on what language you use, described in common workflow language. So that's the wrapper around it that describes exactly what it is. And these tools are pointing to Docker images to be sure that when you pull down that Docker image, you're going to start in exactly the same environment. And if you start in the same environment, provide the same inputs and same configuration, you're going to get the same output no matter when you run it. So when reviewer 2 comes back in 5 months and really has a few small questions that they need you to spend a month on, it's just point and click to rerun. Or if you want to submit this to an agency for validation, they would be able to point and click and understand what you did. On the other side of the story, of course, is as I showed in the beginning, you can use version scripts in multiple programming languages with our interactive analysis. You could use our versioning system. You could use Git, whatever you like, and making sure that reproducibility is there. Extensibility is really important across all the segments that Seven Bridges serves. And here I'm showing just a rough conceptual outline of the Seven Bridges platform, or any one of the platforms that I showed has compute instances and storage buckets, and then it organizes things into projects. So we have a lot of different tools that you can upload data. You could write integrations, you could reuse APIs. And further, we have a tool called Volumes, or Connected Cloud Storage. Now, this was largely developed within the Kvatica project that was really impactful in order to say, we don't need you to put the data onto our storage buckets. You can just leave it on your storage buckets. Or maybe it's not your storage buckets, it's AWS Open Data storage buckets, or a federal agency storage buckets. And they can control all of the security and maintenance. And you just decide, is this read-only, is this read-write? Go through the cloud provider credentials, and then we just see aliases show up in the project. This is really powerful as, data does not necessarily all get replicated to all regions of all cloud providers, right? There is, even within the consortiums we're in, there's clear examples where, okay, this only exists on, right now, the integrated Canine Data Commons only exists on AWS. The GTEx dataset only exists on Google, and trying to figure out, okay, well, how do we do that? How do we avoid egress? Another aspect of the extensibility is we have a multi-cloud functionality, which would allow, as I showed in the beginning, if you imagine these purple things are the projects or the workspaces, we take this data that's only on AWS, process it in an AWS project. This data, which is only available on Google, process it in a Google project, and then combine the results here in an AWS project. That means the only thing that's egressed is in a dashed line here, which is gene counts. For those of you familiar with this, they're very tiny. So there is some egress. It's about 1/10 of a cent in this. Rather than connecting directly the raw RNA-seq files here, you would be in tens of dollars of egress to just pull them from Google to AWS. So we really try to make this flexible, extensible, so that users can bring compute to the data where it lives, and they can connect, most efficiently, and again, focus on the science. The final thing is scalability. Again, like reproducibility, this is really just sort of table stakes for the cloud. If you're going to the cloud, you should have infinite compute and infinite storage, and that's the marketing way to look at it. I'm not going to promise infinity because the engineers will kill me, but we can say that we do have a large pool of diverse instance types. There's about 50 per provider. This has evolved over time, again, working closely with the different users of 7bridges to understand, what are the different workflows that are run, what instances did they work on best. That 50 is on AWS. I believe we have a similar amount on Google. What's been really impactful for people is spot or preemptible instances to reduce costs substantially. And also, there's intelligent queuing. So if someone did come with, I don't know, something obscene, something really high over the amount of that's allowed in their contract or the amount that's allowed by the platforms, it will be intelligently queued so that you can spin up all the jobs that you want, and it will load balance and control costs for you and make sure that things run as you expect. And this is also really important for optimization, having the scalability. So here we're showing instance metrics for Strelka 2, which is a popular semantic variant caller. It's hard to see, but this is CPU, disk, RAM, and it's going from 0 to 100%. So it was originally running in this instance, C4 8X large, and you can see it's not, we're not maxing out the CPU, we're not maxing out the disk or the RAM. By optimizing and choosing the instances intelligently, paired with the 7bridges scheduler that will populate this instance optimally, so parallelize jobs optimally within the instance, we max out the CPU, but we don't go over. Nothing, everything runs just as well. It actually takes 1 minute longer, but because you're processing so many more samples, the cost is reduced, more than 4 times by doing this. This is really important for an individual tool developer. It's also important for the communities we serve. If you think through the funding agencies, for example, in Kids First, they're bringing together XO1 data for large number of whole genome sequencing using a GATK4-based pipeline. We worked together with the bioinformaticians at Children's Hospital of Philadelphia and the bioinformaticians at 7bridges to really optimize that pipeline to get the runtime down to 1/3 of what it was and the cost down to 1/2. And that becomes a really big deal when you have these big reductions, because these agencies are putting together, tens or hundreds of thousands of samples, and you start to add up those little savings per run, such that now that we're getting to a lot of money, and now the funding agency can actually put a lot more data available, and put that money, instead of wasting that on compute, they can put that towards enabling more researchers and enabling more results. So these were the common elements, and as I tried to convey, these were really we built these ecosystems for the users at the time. So the people who are already at universities, already doing cancer research, already developing tools, people in the heart, lung, and blood space, people in the pediatric space, and it's been a great journey, and it's been really impactful for those communities. And the question was, how can we extend it? So when I joined, as I said in the beginning with the cost function derivation, for 10 years before joining 7bridges, I was in machine learning. When I first joined the company, I was working on the APIs, and I was trying to figure out, okay, how do we create a project and put in some folders? All right, we'll write that script, make a Jupyter notebook script and run it. Make recipes and tutorials. Talk to my bioinformatics colleagues in the company, and she would tell me, okay, if you wanted to do RSEM, you would do this. So I translate that to Python, and I write some witty markdown and then publish that. And I was thinking through, like, wow, this is easy. And again, it was 2016, which is about the last time this marketing approach was relevant, was shared with the easy button. I said, it's really easy to do this, and people can just kind of click and run and put in some credentials, and it goes. I wonder if we could get to a larger community. And my best friend is a high school teacher in New Jersey, and talking it through to him. I'm really interested in STEM education, and we brought this to our program officer at the NCI, and thankfully they said yes. We said, can I drive down to New Jersey and see, can we teach high school students this? Can we see if these platforms can be used for education? And they were excited about it, and we were excited, so I jumped in the car and drove down to New Jersey. So I'm going to review what we've done as an online teaching foundation. We started in William Tennant High School. This was 2016. We had 2 lectures. That's me in the purple walking around with all the kids. Ignatio's on the left, and Mark is actually out of frame. But it was me and the 2 teachers. And we had 2-hour lessons, or hour and 15-minute about. It was also in April, so surprised we kept their attention, but they were pretty into it. The first day was much more STEM outreach than data science, but it was really, people were excited. Framing it. It was an AP genetics class. So juniors and seniors in AP genetics. And the first day was, okay, we're really excited about this. We're excited about precision medicine. Do I need to be a clinician, or do I need to be in the wet lab to do that? So we walked through, okay, those are 2 possible paths. There's a number of other ones. Genetic counselors, hardware, analysts, really spanning the spectrum, and got people excited about, okay, what is possible? And we ended the day with talking about the TCGA data set and what do we have on the CGC and what would work and what wouldn't. What was really exciting in the second day, 14 of these students analyzed differential expression in tumor and normal data in TCGA. And that was really impactful for me, of like, wow, they did it, it worked. I used a project and used that for a way to share tools, a skeleton of a tool that they could slightly modify. We used our search technology so they could find the data and be able to provide comments. We used the billing group so that we could do this efficiently. And it proved to be a really powerful teaching tool. And we got some really nice feedback from the students afterwards. It basically saying that this guy, John, he really felt empowered, because rather than saying, you're not old enough or it isn't the time, he just had the tools in front of him, and then he could start to work with this. And full disclosure, no one cured cancer during that day, right? But what I really hope is that we lit a spark in these kids, and that maybe help them change their career path and help them understand what's possible. So afterwards, everybody's excited, high fives. And Ignacio and Mark and I keep talking for the course of a year of, this was great, how do we get this into the syllabus? And this is where we really ran into the first challenge with this model is, yes, it was great, yes, it worked, but this is something that we're kind of like, I was kind of doing in our part-time, he's doing in his part-time, and forming it into the syllabus is a big deal, and really investing that time and developing it. So we really hit a block there of it. It's a good foundational tool, but it's not gonna do it on its own. You need to pair it with technical training. So we took a little bit of time and regrouped, and I wanna talk for the rest of this about the things that we did over the course of 2019, starting with the Summer Institute of Statistical Genetics at University of Washington. So Dave Robertson gave this talk. The idea here was for geneticists to learn statistical methods. And it was about a 4 or 6 hour workshop of some slides and live work. So it was more detailed training than the high school students had. They were obviously more prepared than the high school students. And we had 70 grad students just spin up our studio all at the same time. And for anyone who's given a workshop or a training before, I know we did this back in early 2016. It can be a disaster because we were on a conference Wi-Fi, a hotel Wi-Fi. Everybody was downloading 2 gig Docker images. Steve's version of Java was different than Jane's. Joe didn't have root, and you wind up with spending all this time on configuration. But here, through our interactive analysis, we were able to make sure that we spin up exactly the same thing with exactly the same packages up, have access to all of the relevant data. So students are really starting at a point of, okay, now we can work with it, and we're not going to spend the first half of our workshop with tech support and troubleshooting and crashing the internet. So this was a really productive thing that started off the summer. The next thing was really driven by Alex's Lemonade Stand and Children's Hospital of Philadelphia. They're doing something here, and I have the link in the slide to the Git repo on the Open Pediatric Brain Tumor Atlas. They're actually collaborating here. They had a hackathon last year, and they're working together to collaboratively write a manuscript, which I think is just really awesome, and I'm really excited to follow the progress of this. How Kvatica was involved, it was one of the ways that people could access the data. And here I'm showing a public project, which is like the project that I started with, but this is one where you can put in some description. You can also put in tools. You can put in tools. You can put in tasks. You can put metadata on the files that people could start from if they wanted to understand. Okay, what data is available here? What's going on? And what's really nice is while this hackathon was last summer, this public project persists. So this remains an opportunity for self-guided onboarding. Solving some of the problem of, okay, the platform provides the foundation, but how do users learn to engage with it over time? And this could provide some self-guided examples. Next, Phil Brooks gave a talk at the Data Intensive Biology Summer Institute at University of California, Davis. This is, when I saw the press around this, I was so excited to attend this, but I would be so unexcited to be a teacher here because the tagline was no experience required or expected. And it's people from biology and basic to learn basic BICS. So I think that's a really, really challenging but really interesting problem statement for the instructors. And I think it really well aligns with what was identified by the NIH Office of Data Science. So it was kind of similar to a Cold Spring Harbor workshop. They were able to learn a breadth of tools. The training that Phil gave was a detailed technical training. I think this was approaching about a full day. I mean, these trainings are getting longer as we get through the presentation on RNA-seq and differential expression. And what was nice here is that we generated an entire document that the users could take home and be able to replicate what they learned that day and then have a way for them to build off it and move forward. Dennis Dean gave a talk at George Washington. This was actually one of the first lectures in the semester last fall. It was similarly using the RNA-seq and differential expression. And here he started with introducing them to public workflows. And the idea was that they would create a biocompute object for the midterm. So Dennis gave an entire lecture, worked through the different things, introduced the system, introduced the different workflows, and then talked a lot through the biocompute object, which is really, for those of you who aren't familiar on the call, this was just, I think, 2 or 3 weeks ago adopted by the IEEE as an official standard. The expectation is that within a quarter or two, then the FDA will likely adopt this as a way to describe workflows for the reproducibility, as I said. So if you've developed some kind of diagnostic that requires a bioinformatics analysis between the raw data and the, for example, support or suggested idea, it's very important that the FDA could review through that. What exactly happened? What's the provenance of the data? How is it configured? And the biocompute object is a nice wrapper in order to do that. And it works really well with a common workflow language and the way that tasks are described. There's a number of different people across academia and industry working on this. Seven Bridges has been an instrumental partner in this, especially Dennis has really invested a lot. But what's exciting is, as I said, this just got exposed this just got officially adopted a couple of weeks ago, but we were able to really bring it to students as it was in development and not have any delay between when this thing would be accepted and waiting to develop the materials and then moving forward. It was really, students were exposed as soon as it was ready, and you could really shorten that time to be able to work with these standards. The final one was led by Manisha Ray on the CGC. This was at Georgetown, also in the fall. It started a little later in October, but the first lecture, Manisha was able to say, okay, well, this is how workflows work. This is data. She walked through a really nice example over here of single cell clustering with Surat using bone marrow cells. It's actually kind of similar to TSNE, the way that it's done, but you can see the different categories of cells that get broken out here. And what was different about this one and really exciting is that after Manisha's lecture, there was detailed technical training by the Georgetown faculty for the remainder of the semester. And the students were learning how to create these differential expression workflows, and that was actually part of their midterm, was to actually create it. So it was paired, the powerful platform along with the technical training from Manisha, and then consistent technical training also from Georgetown faculty to really complete the package and really move things forward. So reflecting on these, what we found is that the core elements that we started at in the beginning that really made these platforms useful for the current research communities also made them very powerful techniques to get to the next generation of data scientists, the next generation of bioinformaticians, bringing more people into the platform and onboarding them. So collaboration is really critical, as I hope I've hit on in the talk, for sharing tools and data, giving feedback. It's also the billing groups allow us to efficiently allocate funding, us or the funding agencies, or whoever wants to. Usability is key for diverse learners, bringing in also, very importantly, the industry standards tools. So being able to start from 400 and some tools that are available, so that you're not going into a blank environment and, okay, I have this powerful system, I don't know what to do. But no, okay, if I'm doing something on image processing, I can already start with this tool that's doing a semi-supervised clustering of gray matter, white matter, CSF, and tumor. And I could build from there, because the thing I want to build is near that. Additionally, the guided self-learning, as I tried to show with the public projects, enables us to scale a little better, where we're not consistently having to give the course or have professors give the course, but to be able to have materials. This could be the public projects. This could be webinars. This could be FAQs and quick starts and things like that, to be able to do this. Reproducibility is really important. Obviously, I would love if everybody started in a 7 Bridges platform and then went off to industry and brought 7 Bridges with them and just kind of 7 Bridges complete life cycle. That's not realistic. If people need to go to another environment, that's totally fine. What's important is that the tools and things that they developed here are portable and can be reused. And one of the easy examples of why is some data has locality restrictions, that it can only run locally. Well, that's okay. You can then take your workflows, and in some cases, you can run them locally and get the same result. Extensibility is really important for custom analysis. Obviously, if you're learning data science or bioinformaticians or bioinformatics, we don't want to create an airlocked, totally locked, closed down environment where you only have the data that's there and you only have the tools that are available. You need to be able to bring in your own data, bring in your own tools, adapt them in different ways. And APIs are really key, depending on what's being interacted with. Not so much for training so far, but when you think about interacting with a LIMS system, downstream production systems, it's very important. Finally, scalability. As I said in the UW example, it's really impactful and helpful to have appropriate and stable resources. It's also impactful to optimize the usage, such that funding agencies and researchers are able to make their research dollars go as far as possible and really get towards publications. And the final thing is that public data is free. So different institutes and centers have invested to keep, for example, 1 copy of TCGA on AWS. When we connect to that and when the user is authorized to access that on the CGC, we're actually not copying that data, but we're making a pointer to it. That means that people can analyze that data, and the raw data is not charged. They don't have to store that. They're not making their own copy of TCGA or of different data sets. And they're only dealing with the compute cost and the download and the derived data. And to put some context around that, if you remember the high school example with the 14 people developing a tool and running some quick analysis, the total cost for that, I think, was $12 or $15. It was extremely cheap, because just being on the system and exploring and understanding the data doesn't cost anything. Changing tools doesn't cost anything. Similarly, for the 70 people running our studios at UW, and doing association tests, I think that was on the scale of a few $100s for running that. And obviously that's more than the high school, but thinking through different options of making that many exactly stable systems available over time would be a much larger investment than that. So it's actually quite economical to work on these platforms for the research community. For take-home messages, we're really grateful that the NIH institutes and centers have made foundational investments in these ecosystems, and it's been really empowering for current researchers. What we've found over the last few years, and what we're excited to continue going forward, is how powerful these platforms are as teaching tools to connect trainees to actionable data. They really provide a very solid foundation for teaching the next generation. At the same time, we understand that technical training is an integral component here. So you can't just have the platform as a foundation and then not guide people of what to do. So we found that's really important, whether that be Seven Bridges staff or university staff or self-guided onboarding and things. So those are the 3 points. We really think these platforms can be an exciting component of teaching the next generation of data scientists and bioinformaticians. Might ask for you, if you think this fits either for your existing research or for teaching that you want to do, please reach out to the link right there. That'll go to a bunch of us, and we'll find the right person to get you in touch. And I hope we have some time for lively discussion. Okay, I see. Just a reminder to everybody, if you had questions, you can put them in the chat. I see the one here that I think will be interesting to discuss. There were a lot of RNA-seq gene expression functionality on the slides. What genetic association analysis workflows do you support? e.g. BOLT, LMM, SAGE, PLINK, LSSD, polygenic risk scores, clinical outcomes, time to event analysis. That's a great question. Yes, a lot of the things that we were talking about were all focused on RNA-seq. That was really trying to reproduce and make stronger the training module that we had. But we do have a lot of association analysis. One of the ones that we're working on with the Data Coordinating Center at the University of Washington is the Genesis pipelines for GWAS. There are polygenic risk score things on there. We have PLINK as well. We also have non-genetic pipelines that are on there. As I mentioned, we have imaging. There's some time series pipelines, different things. Another question. Sorry, I went through on my video. It's weird not to hear and just read questions, but I'm going to go through them. Let's see. This is a good one. If I have a new trainee who hasn't been through strict NIH Data Commons vetting, can I give them access to a subset of the data in my restricted project so they can start training? No, you cannot do that. But one of the things that we've really, like if it's controlled data, you cannot add them to the project and suddenly they will be able to work on that data with you. We actually have technological things in place to block that, and there's also policy things in place that you would not be able to do that, not from Seven Bridges policy, but from dbGaP policy. However, one of the things we really focus on and we really continue pushing is on having access to open data, because this is a common blocker, right? A new grad student comes into the lab, they want to get started, they want to learn the tools. Oh, they need to get fully approved and they can't. Well, this is, having that open data, having 1000G up there, having imaging data sets that are uncontrolled is really important to be able to do that. So we try to make it as easy as possible for them to go forward, but we strictly maintain the authorization requirements for the data sets that we have access to. Another one coming in. Okay, what sorts of public data sets and what data types can users get access to from the Seven Bridges platform, and how does it make it easy to do so? That is a great and difficult question. I think I have an extra slide on that. One second. So here are some of the data sets that are available on the Seven Bridges platform, on different Seven Bridges platforms, excuse me. So depending on which platform it is, what data would be available. On each of them, we also have a sort of data overview that you could look in and start to see what's available. I actually can try to stand that up right now. Live demos are the best demos. And try to show you an example of how that works. One second. But it's a really important question because it becomes difficult to understand, especially for data sets that have a particularly lengthy application process. Is this worth my time, right? Like that's an issue. Should I be attempting to get access to this data, or am I going to go through the entire process and then find out that it really doesn't have the particular data that I need to answer my research question? So here, I'm sorry, I realize I'm not sharing that screen. I'm gonna switch over to the CGC quickly. Here's the Cancer Genomics Cloud dashboard that's coming in. And you can see we have this thing called Data Overview, which would allow us to pick all different data sets that we could potentially look at. And here's TCGA. And you could start to look, okay, what is available in, I'm interested in leukemia. So what's available there? And then it responsively will tell you it's about balance between females and males. The age peaks in the 60s. These are the different types of samples. And for it, the most common experimental strategy is the methylation array. There's no whole genome seq available. And you could work that way and try to understand what techniques were used, or for the formatting question, you could come back through this way and figure out the types of files, or I believe category is going to get me to the approach as well. Yeah, CNVs, transcriptome, different things. Okay. There's a philosophical question here, and I love philosophical questions, so I'll try. What scale does the working in the cloud become necessary? What problems does a cloud platform solve that aren't addressed locally? It really depends on what your local capabilities are, and also how much collaboration you're doing, right? So if you're a one-person army and you're working with a small amount of data, or you have an infinite HPC and you're just going to do all the analysis start to finish, it's probably less necessary to go to the cloud. As the analysis gets more complex, as you don't want to get into a situation, we talked to numerous people over the years of, we really want to work on this platform because I looked across my institute and I noticed that we have 7 different copies, or 17 different copies, or 47 different copies of TCGA all stored on our local cluster, right? Like that's just a huge inefficiency to do that. There's also the issue of, if I'm collaborating with someone, I think we've really seen this as we've all gone into lockdown and quarantine. Okay, I can't get into the institution. The VPN's not set up to get me to that cluster so easily. How do I leverage these resources? How do I bring this together? How do I do the work? And that's that comes up as really important as well. Other aspects is some data sets are only available through the cloud. So maybe it's not through one of our clouds, but it's available through a different cloud. And do you want to spend the time downloading and storing all that data, paying for the egress of all that data? Or do you want to say, hey, this is up in Google Cloud West, I'll set my project over there, and then it transfers almost instantaneously, because from Google's perspective, it's going from Google West to Google West, and you don't actually have any egress or time wasted waiting for it, or storage of the data. So it's a much deeper question. I think it's a topic of another webinar, but it really depends on your use case and what capabilities you have locally. I see one on some additional questions about the multi-cloud stuff. I do want to say, I'm sorry, that in the middle part of the talk, when I was trying to give examples of the core functionality and the features behind them that have proved useful to existing users, it was really fast. I didn't have time to dig into each of them. Probably each one of them could be their own webinar. Again, please hit that contact link, and I'm sure our team would be delighted to develop more of these as freestanding content. But given the time constraints of what I wanted to cover here, we couldn't get into full details. Sorry about that. But I hope to see you at a future webinar. Another question. Can workflows or interactive analysis, example, Jupyter and RStudio, developed on the platform, be run locally as well? Yes. So if you just take a script and you take all the data and you have a copy of the data locally, it will run the same if you give it the same configurations. There are some caveats in terms of, you're running in the cloud on a advanced GPU instance and you bring it locally and you just have a regular standard compute instance. It potentially will not run as quickly it may not run at all if you're hardwired to some dependencies of NVIDIA drivers. But overall, things that are developed in the cloud should run locally and should generate the same result. It may take a different amount of time to get to that result. In terms of the workflows as well, I mean, we're working with the VA and within the Initial BI project and the Million Veterans Program to actually do exactly that, to take some of the associations tasks and the variant calling workflows that we have and to run them locally, because there's data there that actually can't move up to the cloud based on the consents of the veterans that provided it. So for the data that can go to the cloud, we're computing in the cloud, which is great and very efficient for the top med data. And for the data that can't go to the cloud, we're leveraging local resources and computing there, and then bringing together the results and as permitted to answer the research questions. There's a question about, oh yeah, this is a good one. So I only showed a few languages. For the version scripts, what languages are available? So the interactive analysis, we have R, we have Python, we have Julia. There's also an RStudio instance. C++, C#, these are all, you can code in these in the tools and workflows. We don't right now have an interactive analysis that works with them that I know of. Additionally, there was a question about SAS. SAS is something that we're really excited about, that there's a number of people having conversations to make this possible because a lot of researchers are interested in this. I would say that's ongoing to get those capabilities onto the platforms. It's not available today, but it's very much something that we're interested in. There's one more question. One more question about the STRIDES program. So this is a program created by the NIH to be able to make cloud compute and storage more economical for researchers in partnership with Google, with AWS. And is that available, and how does that transition onto the Seven Bridges Programs platforms? I'm excited to report that users of the AWS, for AWS on both Kvatica and for Biodata Catalyst, are able to leverage the STRIDES discount based on that they're being NIH-funded researchers. So we'd be delighted to talk to you about that. Oh wait, late question, 5 minutes left. What kind of bioinformatics professional services does Seven Bridges have? And some examples of the kind of work our team has done with customers. Don't quote me on the exact numbers. I think we're roughly about 300 people as an organization. About 50 of them are in bioinformatics. And I think it's about 20 of them have PhDs. We also have a large scientific component in the partnerships team and in the programs team. So it's really spanning a lot of different depths of expertise. Again, from a machine learning background, I was delighted yesterday when NLP came up and people were just like, hey, do we have any apps for this? How does this work at Seven Bridges? And somebody jumped onto the Slack and said, oh yeah, I got my PhD in that, and I have these tools. And so it's pretty varied, but it's, I would say, a good depth. Some of the work that we've worked on with our customers, again, through the foresight and generosity of the ICs, we've created these different collaborative projects in Kids First, in the Cancer Genomics Cloud, in the Cancer Research Data Commons, and in the Biodata Catalyst. And it's really led to some really impactful things in terms of, rather than getting pilot funding, which I definitely should have talked about a few slides ago, but I forgot. So when you sign on, you get approximately $300 of pilot funding to get started, so that you have enough that you don't feel like, okay, I'm afraid I'm going to spend all my money, I don't know what to do. You actually have enough to onboard and get started and run some pretty decent analysis, given that the data is free in terms of storage. But this is cases where the ICs have provided even more funding and often partnered with someone at Seven Bridges, either on my team or on the bioinformatics team, work to develop tools together. Some of these, for relatively small amounts of money, like $10,000, $20,000, have yielded two publications, one under review, and one near submission. In terms of our bioinformatics team working to optimize the tools or working to add one tool to their existing workflow. So that's certainly possible, and that's been really fruitful because it enables our people to stay involved in the science. It really aligns the incentives really well of they want to solve this problem, we also want to solve this problem, we're collaborating together. And in the end, it also generally, not in every case, but generally it benefits the broader community as well. So these tools that get developed, when they publish them, they're excited to open source them, or they're excited to share them with the community. And the licensing choices they make, Seven Bridges is not involved. We're happy to, we definitely uphold whatever they decide. But for the many people who open source them, then we can bring them right back to the Cancer Research Data Commons and say, okay, we have this great new tool for microRNA analysis that we didn't have before, and everybody can use it. And again, pushing towards the end of that product to market or public research to publication lifecycle of, you already have this great foundation and things to build on, then you can add to the end, focus on the area where you'll have the most return on investment and leverage the tools, stand on the shoulders of giants, pick your analogy, and you can do it. Okay, looking at the chat, we got through all the questions. Thank you so much, everybody, for attending. I really appreciate your time. Please reach out to that contact list if you want to talk more, learn more. I hope everybody stays safe, wears a mask, and hope to see you again sometime soon. Thank you so much. Bye.